Okay, hello, welcome to a new topic on Mr. Gill's Science Stuff. So this is variation, right? So we're going to start with a really small, we're going to move up to the massive. Now this panorama picture here I took in Fiji on the Natawa Peninsula, and it made me think massively about, think of all the levels of variation that there are in this picture. From the absolutely enormous, that is the largest seawater bay in the South Pacific, right the way down to the tiny, to all the little invertebrates that live in those forests, right down to the genetics, right, and all of the different levels of variation that have to have come together to have produced that. Okay, so that's basically what we're talking about in this lesson. So we'll move straight on. As you can see, look, in this topic, okay, we're going from the DNA over here, right, up there. So we're going from that. Now you should know how DNA works already from previous lessons on transcription, translation, protein synthesis, so on and so forth. You should also know a little bit about chromosomes. So we're going to talk about, in this lesson, chromosomal abnormalities and chromosomal problems that lead to variation. Okay, we're then going to move up through this topic up to talk about how chromosomal variation, meiosis, the splitting of chromosomes, leads to species variation. Now we all know, right, that those are both dogs, okay, but they show huge variation, massive variation. We're then going to move up to habitat variation, habitat variety. This idea that you've got variation within ecosystems, and that's a picture I took from the top of a hill in the Ecuadorian cloud forest. Incredibly diverse and weird environment. And then we've got this one down here, moving up to the whole Earth as an ecosystem, and conservation, and the idea that we need to preserve biodiversity and preserve variation for our own good as well as the planet's. And obviously I didn't take that photo. So our first lesson today is on mutation. So how DNA changes over time, how we generate new alleles that then become new proteins and various other things. Okay, so what I want you to do first, because I imagine mutation is a fairly loaded term, you already know a little bit about that. I'd like you to make a list of as many things as you can that involve the word mutation, or that cause mutation, that you can think of. I don't really mind how tabloid you are with this, because this is often lumped in with things in the tabloids that cause or cure cancer, or that kind of thing. Please don't go full Marvel universe and start talking about radioactive spiders, because that's not what we're after. That should be fairly obvious, you're A-level biologists. But then I want you to rank them in most serious to least serious. So pause the video, take 5 minutes to do that, go. Okay, lovely, moving on. So this is the bit of syllabus we're looking at, the idea of what type of gene mutations we're talking about. We're talking about micro mutations, so substitutions and deletions, the idea that they might have absolutely no effect whatsoever, but they might cause a change in a protein which can change a phenotype and change what a cell does. We're also then going to look at chromosomal abnormalities, nondisjunction, and all that kind of stuff that can also lead to variation as well, in a different way. So let's start with the definition, because it's always a good place to start. A change in the sequence of DNA bases that alters the protein product is a good definition of what a mutation is. Now you might be thinking, well hang on, there's loads of DNA you've already said in other lessons that doesn't code for anything. So therefore, aren't there mutations that won't change the protein product? Yes, absolutely there are. They're called silent or neutral mutations. And yes, they do exist. But the ones we really notice are the ones that change the protein product. So let's look at an example here. So this is a frog. Alright, so let's say the frog suffered from a mutation in one of its cells, its chromosomes. Now the most likely thing that mutation, that alteration would cause would be nothing. Okay, neutral or silent mutation, so no effect on the organism. One, because of all the non-coding DNA, both outside and inside genes, so all the non-coding stuff outside and the introns inside genes. But even if it was in an exon of a gene, then the code's degenerate, remember? Right, the code, the third base isn't as important as the first two, so it might change one code onto another one that codes for the same amino acid. So then the next most likely thing is that a harmful mutation might alter the survival capacity of the organism, and after that then you get the possibility of a new useful allele popping up which could generate something better in a particular environment, which would then lead to evolution and natural selection. Okay, so hopefully you got your causes of mutation. Okay, the idea of mutagens and various things in the environment that can cause things to mutate. But I want to tell you that the most likely thing to mutate DNA is actually just random chance and spontaneity. Okay, so obviously throughout your life, huge amounts of DNA in every single one of your cell has to be copied, used, changed, so on and so forth. Okay, so mutations may occur randomly or spontaneously from just errors in replication. They may also be induced then by environmental factors called mutagens, and I imagine that's where most of your list sort of focuses. So we've got down here ionizing radiation, UV rays, X-rays, beta, gamma, all that kind of stuff. We've got, I think back to GCSE physics, we've got viruses and other microorganisms. A really good example of that would be HPV, which if you happen to have a cervix, you might have been vaccinated against, because of course if the virus lands on a mucosal membrane like that, then it will basically cause the cells to have to constantly repair and replace themselves, which can increase the risk of cancer. Okay, hence vaccination against cervical cancer. You've got carcinogens, things like asbestos and various other environmental poisons and irritants and so on and so forth that can cause cancer. That's how it's reported, but they have the energy to alter DNA and change mutation and change your DNA. And then alcohol and diet, of course, are lifestyle things that we all do. So loads of things in there we know, but of course about a year or so ago, there was, maybe more, there was the report that that cured meats, so things like bacon, sausages, various other things, can increase the risk of bowel cancer by causing mutations. Okay, that's obviously a hard pill for me to swallow because I love a bacon sandwich, but unfortunately that is the case. It will increase the risk of bowel cancer, so I've had to cut down, and obviously I recommend you should do the same as well. Right? So moving on, I would be surprised if anyone didn't get this. This is probably the number one thing that will come up on your list, which is tobacco tar. It's one of the most damaging constituents of tobacco. Smoke causes chronic irritation of the respiratory system. It's a major cause of cancer in smokers. Contains at least 17 known carcinogens. So if you take nothing from this lesson else, it's don't smoke. Okay, and that's before we even get to all the chemicals in the skin, in the wrapping of the cigarettes that are even more carcinogenic. And I want to draw your eye down here to benzene, which is a chemical we struggle to store with all of our experienced chemistry technicians and all our experience in our equipment in our laboratories here. So that's because it's really, really dangerous. But you can of course go to a newsagent, buy a packet of cigarettes, contain a load of this stuff, stick it in your mouth and set fire to it, which I think is madness. But there we go, that's the situation we're in. So don't smoke. So moving on then, so that's the causes. Let's move on to stage 2, the types of mutation. Substitution, deletion mutations. They are exactly what they sound like, okay? There's no trick to it, they're exactly what they sound like. Now for this part of the lesson, you will need a genetic code, so you can either screenshot this one, or click on the link in the description to a genetic code, or pick one up from a textbook or whatever it is, because I'm going to ask you to do some transcription and translation, which you will be asked to do in an examination. So let's move on then. Substitution is literally just changing of bases. Okay, it's substitution. You've got a load of bases in the genetic code, in a DNA strand. You take one out and you put another one in. Now remember, that can have no effect because of the degenerate nature of the code. The idea that it could be CCC to CCU, which both code for proline, same thing, right? But it could cause a change in one amino acid. So it's where one base is paired incorrectly. The effect of this is it alters one codon. It might not, it might do nothing, but it may well change one amino acid. But one thing it won't do is it won't affect the rest of the chain. Everything upstream and downstream, everything before and after that mutation will not be changed. It'll be that one amino acid maximum. Okay, we call that a point mutation, and that's an important key term. Okay, so it's a point mutation. So let's crack on to an example then. So this is the same DNA strand we're going to use for all our examples. So once you've transcribed and translated it once, all we're going to do is mutate it, tweak it to illustrate these processes. So what I want you to do now, get your genetic code out, right? Basically take that DNA strand up there, turn that into an mRNA strand about there, and then work out what would be under these white boxes, which are the amino acids. So pause the video, crack on now. Okay, good. So hopefully you got those RNA bases, that's your mRNA there, and you got these amino acids underneath here. Methionine, tyrosine, lysine, glutamate, proline, cysteine, alanine, glutamic acid, and stop. Now there's no way that you will be asked to remember these codons and these amino acids and what they stand for. You'll be given that information in an exam. I always get confused between GLU and GLUN, your glutamates and your glutamic acids, your glutamines and your glutamic acids. I may have done it there, right? But you always get given the information you need to answer the question. So as long as you can use the information, you're doing great. Okay, so let's start by introducing a substitution mutation. So please notice this TTT up here. We're now going to introduce a C into the middle of that. Now I want you to now take a moment, should take you a minute to work out what the consequence result of that would be. Okay, good stuff. Right, so there's our original strand, right, what we're aiming for. Right, what we actually end up with is an alteration of an A to a G in our RNA strand, okay, and that leaves us to, these two are fine, but our lysine is replaced with an arginine, and then the rest are fine as well. Okay, so that is our point mutation. Okay, so that's our point mutation. So we've changed one amino acid and not the rest of the strand. Okay, good. So an example of this, right, one really common genetic disorder caused by this point mutation is sickle cell anemia. Okay, and we know quite a lot about it. It's right here on chromosome 11. This is the hemoglobin B gene. Okay, and what it does is it codes for one of the two constituents of hemoglobin, right? And if you've got this single base change, it changes a single amino acid, that lovely sort of tertiary structure that contributes to the overall quaternary structure of the hemoglobin collapses in on itself. That in turn makes the hemoglobin collapse, which in turn makes the blood cell collapse from this lovely biconcave disc shape through to that sickle shape there. Okay, so that is what happens. Now that can obviously be quite bad. If you've got one copy of that allele and one copy of a working allele, half your blood cells will be sickle shaped and they get stuck in your smaller blood vessels and lead to this sickle lesion that is up here. And you might think that's really bad. You can't carry oxygen around, the blood's getting stuck. How does this survive in the population? Well, not only in certain parts of the world does it survive, but in certain parts of the world it's actually the dominant situation is to have one copy of this gene, because there's an association between this and malaria. So in malarial areas, it's actually an advantage to have one sickle gene. And that's a brilliant example of modern human evolution. So you can go and look into that yourself, because that's not only another lesson, it's also a whole other topic that we're going to come to at another time. But there are other videos on that on the channel if you want to have a look. Right, so move on. Deletion mutations, again, are exactly what they sound like. They're basically where one base is missed out. So you've got bases strand like that, take one out and all the rest of them just get pulled together. So what happens there, the effect of that, is it basically drags all subsequent bases back. Because remember, the ribosome in translation is going to go along reading in threes. It's a triplet code. Okay, if you've taken one base out and all the others are moved up, then the threes that it reads together are going to be offset by one. It would be the equivalent of taking the first letter off a word in a sentence, right, and then shunting all the other letters backwards but keeping the spaces in the same place. So the first letter of each new word becomes the last letter of the previous word. Okay, so we turn everything after the mutation into complete gobbledygook. So because it's changing the reading frame and the way the ribosome reads, we call it a frame shift mutation. The previous was a point, this is a frame shift. Okay, so I'll give you an example here. Look, we've got our A question mark G there, that's our deletion. So work it through, what's going to happen, pause the video, work it out now. Okay, so it's really pretty obvious what's going to happen. There's our original strand again. Our mRNA, I've tried to keep the codons that should exist coloured in the same way. And here's the reading frame. This is what the ribosome's doing. So up to that point, so far so good. But now we're reading UCG together. And then, right, all the rest of them are read in the wrong way. They're offset by one. Okay, now the amino acids up to the point of the mutation will be exactly the same. After that, you can see all different. So massive problem. You see the two question marks at the end there, it's because we've broken the stop codon. Now if you remember the transcription lesson in the previous module, right, that means that the ribosome isn't necessarily going to know to stop, and it will run on and on and on off into what we call the poly A tail, if you remember that, which means it's going to run off into a region that's rich in AAA, so therefore the protein is going to be rich in lysine. And that's something, because you can see here, look, AAA codes for lysine. And that's something that you often see as a result of frame shift mutations, is lysine rich proteins that don't work, don't do their job. Not always, because the deletion mutation could generate a stop codon elsewhere, stopping the ribosome early. Don't know, possible, right? But lysine-rich amino acid chains, proteins, are often associated with deletion mutations. Okay, so let's look at an example, probably the most common inherited disorder in the UK, by a long way, is cystic fibrosis. It's the deletion of one whole triplet, removing one whole amino acid. Okay, so it is a deletion, but it's 3 bases. So here's a question for you: is it a point or a frame shift mutation? And the answer to that one is that it is a point mutation. Right, because yes, we're deleting, but we're deleting in 3s. So it just takes an amino acid out and leaves all the rest alone. Okay, so when the ribosome's coming along, all it basically is effectively doing is skipping an amino acid. Okay, so it is a deletion mutation, but it also counts as a point mutation. Now, I'm not going to go into what cystic fibrosis is, because you can look back at the GCSE lessons for this, because we covered that quite extensively. But it is common, affects the lungs, affects the gut, affects the movement of chlorine and the buildup of mucus. Okay, so moving on. I'm just going to mention addition quickly, because it is really easy when you know about deletion. It's just the opposite. Okay, the idea that you add an extra base to the coding sequence doesn't take a genius to work. That's going to, instead of pulling all the amino acids back, it's going to shunt them all along. That is a frame shift mutation. Okay, and let's work through an example here very quickly together. I've shoved an extra C in. Of course, that's going to shove an extra C into the mRNA. I've tried to keep the triplets the same. So there's what we're aiming for. There's our reading frame. As you can see, everything downstream of that mutation. So all those are fine, but everything downstream of that mutation is different. Okay, so another frame shift. Pretty easy. Right, the location of these mutations, of course, stands to reason, really matters. So here's 2 broad families. You've got the gametic or germline mutations, which happen in the gametes, and they obviously cause a very serious effect, because if the gametes that formed you had a mutation in them, then that mutation would be inherited into every single cell in your body, because obviously all of them have grown from that one gamete. And that includes your gamete, so you can then pass that mutation on down the generations. Okay, so gametic mutations are very serious, very large consequences. Somatic mutations, however, right, occur in one cell. Now, when that happens and which cells grow from that cell affects how serious that is. In this example here, look, it's quite early on, and it's led to like a patchwork of different pigmentation on that child there. Right, but it could be that it's just a single cell in your skin, for example, gets hit by some UV radiation, mutation occurs that means it grows out of control. Okay, and that then leads to a patch of cells that grows out of control without stopping, acquires new mutations, and that's where we get our links to tumours and to cancer from. Okay, but that would not be heritable down the generations. Okay, so that's the difference between a gametic and a somatic mutation. Okay, last section of the lesson today, this is where we're going to stop after this bit, is the idea that mutations and abnormalities can also occur at the chromosome level, called aneuploidies. Now, you are a diploid organism. You should know that. You've got 2 copies of each chromosome, one from your mum, one from your dad. That happened because when they made their sperm and egg, their 46 chromosomes split into 23, into sets of 23, so 1 from each pair, to make the sperm and the egg that eventually made you. Right? Now, if I think that relies on these chromosomes splitting up properly. If they don't do that, if at some point so a centromere doesn't break and you end up with these things stuck together, you get a thing called nondisjunction, where maybe you end up not with 23 and 23, but with 24 in one gamete and 22 in another. And the result of that, if that 24 chromosome gamete meets a 23 chromosome gamete, fertilization, you end up with 47 chromosomes, not 46. Okay? So we've got triplets here, for example, at chromosome 18, chromosome 13, chromosome 21, due to non-disjunction. The most famous example of this is, of course, Down syndrome. But there are many others as well. Okay? So aneuploidy may involve lots of what we call autosomes, the normal body chromosomes. Down syndrome, possibly most famous, and this child here is displaying a normal appearance of a Down syndrome newborn. And that's the triplet at chromosome 21. Then you've got Edwards syndrome at chromosome 18, Patau syndrome at chromosome 13. Or you can get aneuploidies of the sex chromosomes. So having 2 X's and a Y, as opposed to 2 X's or an X and a Y, is called Klinefelter syndrome. And having 1 X and nothing else is called Turner syndrome. And these are going to affect secondary sexual characteristics, fertility, and so on. But as I say, the most famous one is of course Down syndrome, a triplet at chromosome 21. Right, the idea here, look, chromosome 21 is a triplet there, and you can also tell from these pictures, this picture here, this is a male because there's XY chromosome, so this is a Down syndrome suffering boy. And this is strongly associated with maternal age, the age of the mother. So 1 in 800 births to women giving birth at 30 to 31 years of age comes with Down syndrome. Okay, so it's associated with increasing maternal age, increased risk of Down syndrome, and it's the most common form of aneuploidy in human newborns. Okay, right, so that's us for today. Next lesson we're going to talk about how we actually generate the variation in gametes, generate the variation in chromosomes through the process of meiosis, right, that leads to the variation within an individual, within a species. So the next couple of lessons are going to be on meiosis. I hope you can join me then. See you then. Bye-bye.